fighting today. So these viruses are distinctly different in their genetic makeup. So what drives viral evolution? Why is it happening? Because coronaviruses are known to be fairly stable. So the reason it is happening so fast, plasma therapy. Now, every day, if you open Twitter, you would find calls of people asking for plasma, plasma, plasma. Although WHO started a trial, they later on, said that plasma therapies did not work. That's fine; it does not work. But what it does is it changes your viral makeup. So this is a article in Nature that has come out in April. I mean, it's a very recent paper. It says the SARS-CoV-2 evolution during treatment of chronic infections. This is a patient who was in the hospital for more than 100 days. Ultimately, the patient expired. What the scientists observed, this is from the abstract, that after convalescent plasma therapy, we observed large dynamic shifts in the viral population with the emergence of a dominant viral strain that contained a substitution in the S2 subunit and a deletion in the S1 N terminal of the domain of the spike protein. And they conclude that these data reveal strong selection on SARS-CoV-2 during convalescent plasma therapy, which is associated with the emergence of viral variants. So the reason we need to be very sure that this patient needs plasma and do not, just like antibiotic resistance, what we will be end up are 1,000 strains of viruses which will be resistant to the existing antibodies. So you would be knowing that now doctors are telling that people who are vaccinated cannot donate plasma. The reason doctors are telling this is because if you donate plasma after being vaccinated, you risk the population with a viral antibody, vaccine resistant strain of the virus. But overall, these therapies should be used with caution, which I don't see people doing that. Another reason why we are facing this dilemma is wild animals. We started to think that this virus, COVID, the SARS-CoV-2, affects only humans. So that was only the human to think, because we tend to think of us like a superior kind of animal. But it's not like that. There have been reports of multiple animals getting infected with this same COVID. Although they might not have those severe symptoms, but what they do is they drive mutations. They are the primary drivers of mutations. And this is a paper, this is a blog that has been maintained by Robert Gary, where he continuously studies. So this picture basically shows how many mutations we have seen just coming from animal, human to animal, and again animal to human. So there is a zoonosis, then the disease moves from humans to animals, and then again they come back to humans after infecting the animals. And every time there is a host jump, there is a huge amount of hyper mutations going around. And where would you think the mutations would happen? Obviously, at the spike protein region. And why would the spike protein be the most dominant region? Because it basically allows the virus to select a new host. So basically, every time you have a new host, you have a new variant of the spike protein. And there are multiple research papers which talks of susceptibility of ferrets, cats, dogs, and other domesticated animals to SARS coronavirus. Very few countries are testing this. I don't know in India if any scientist has ever tested a dog or a cat. We have so many of them in and around in our society. Are we testing them? No, we are not testing them. Do we really know what's happening in our backyard? No, we don't know. So these studies are basically from China. There is a study from Netherlands. This is on a farm minks. So in Netherlands, there was a mink farm where the virus jumped from humans to minks, and again from minks to humans. But thankfully, this mutation has now reportedly died off, because there was a lot of culling of animals. Most of the farms were liquidated, and somehow the virus went out of the population. But there is always a risk. So domesticated cats, this is from US. And now, the most important, why these mutations are very dangerous. Now, this is a reprint, and this is not accepted, this is not peer reviewed yet. But this is a paper that tells us that although the Balbc mice, so these are the Balbcs are the common lab mice that we see, the small white ones. So these mice earlier were not getting infected with the virus because they needed the human ACE2. But strangely, the newer variants, primarily B1351, if you see, these consistently have a higher amount of infections or in the lung of the rats. Again, you see this is per gram of lung, how many of plaque forming units they have found. So these results are really dangerous to say the least, because now these variants of of the virus do not really need a human ACE2 to infect, which means this is enhancing transmission to other animals. And once it happens, there can be a chance that there will be antibody evasion. So think of COVID spreading to entire animal kingdom, and now you have a virus that jumps from animal kingdom to humans, from humans to animals. And now it is not dependent on human ACE2. It can bind with cat ACE2, it can bind with rat ACE2. All mammals, all animals producing ACE2 are at a risk. So what happens during these events? The two events that is classical events happen is antigenic drift and antigenic shifts. So briefly, so antigenic drifts happens when there is a mechanism of variation. So what we are seeing is antigenic drift. Mostly it's antigenic drift that gives. So it gives into multiple viral strains, but there is only a minor antigenic change. So your vaccines would work. But obviously, this occurs very frequently, and that's what we are seeing from multiple reports. It is fairly easy to treat. But what is the most dangerous thing? And now it might happen. Now we are on the brink of an antigenic shift. So once there are multiple, host change, you will have two viral strains, and these two viral strains. So when we get infected, suppose a man walks into an hospital, so he gets infected by a viral strain X. So strain X of COVID, he gets infected, maybe in a vaccination center, and then on the same day, while coming on a public transport, he again gets infected by strain Y. And in between, and what happens is when this person gets sick, X and Y recombines, and it produces an entirely new strain of the virus, and that can cause a major antigenic change. This happens very rarely, but what you see, these triple mutants and all these mutants are probably antigenic shifts, and it is very difficult to treat with the existing set of vaccines. So this is a classical study. That is, it's now a citation classic almost, because this has what happened in Brazil in the past few 3 4 months. So in Brazil there was the first lockdown, much like ours. So there was a mandatory use of mask. Then there was a physical distancing eased. Again, strangely, they also had elections. Entertainment venues opened up. Then there was a second lockdown. What you see is in the red line are the COVID-19 hospitalizations. Just like India, the first wave saw far less hospitalizations, but the second wave saw much higher hospitalization rates. And this is despite the fact that in Manaus, Brazil, there was a very high seroprevalence. So they did seroprevalence studies, much like India. We did seroprevalence studies and claimed that, okay, we are immune to the virus and nothing can happen, nothing can go wrong with us. Brazil also did the same thing. I think around the world, all countries are doing the same thing. They're taking the virus far too lightly. and relying too much on immunity and herd immunity. We have not reached herd immunity. We cannot reach herd immunity against a virus which is continuously changing. So this is a continuous fight that we have to fight. So in Manaus, Brazil, what we see is this is the region in December 2020, January 2021, there was the emergence of the Brazil variant of the P1 lineage of SARS-CoV-2. And once this lineage started, the entirely highly seroprevalent group of people again got reinfected. And now, what is the strain status today? Today, if you see after October of 2020, continuously we are getting reports of newer strains. These are emerging lineages. B1617, this is the Indian variant of the virus. So very soon we would be having many more variants. It's pretty natural to have it. So what are the now the variants of concern? So there are B117 of the UK strain. So what happened here is, again, there were mutations, 17 mutations. Again, there was a mutation in the spike protein. Thought to have 40% increased transmissibility, increased virulence, although there is little concern as far as vaccine efficacy is concerned. We have the South African strain. There are 21 mutations, including 3 on the spike. And in vitro studies suggest there is a potential for immune escape. Again, this came in late December in South Africa. We have the P1 variant. We just discussed about the Manaus case study. And here there are 17 amino acid changes. Again, there are 3 mutations on the spike protein. There is a ORF1b deletion just outside the spike protein. And what is most dangerous is there are anecdotes of reinfections, and there are potential of immune evasion. So we are continuously seeing there are multiple strains. There are US strains, Midwest variants of the US. Now US, and we have the Indian double mutation strain. But a strange trivia is people call it double mutation, but actually it has 13 mutations in the genome. It was discovered in 5th of October 2020, and we kind of took all these things pretty easily, and today we have multiple double mutants. And basically it is called double mutant because in the spike protein region there are 2 mutations, 3 mutations actually, and they are of great concern. Effects of vaccine efficacy are under investigation, but preliminary reports again say that the vaccine is working. Even if the vaccine is working, we understand the history of coronaviruses. We understand that humans are repeatedly reinfected with coronaviruses. If you talk of endemic coronavirus like the 229E, OC43, HQ1, and NL63, a typical person gets infected by these viruses every 2 to 3 years, and these are scientific studies. So it's no wonder that we are seeing reinfections in case of COVID. What a back to the future experiment a few people studied. Again, this is not yet published. This is taken from a preprint server. But what they did is this group of scientists, what they studied was they took archived human sera from 1980s and 1990s that had neutralizing titers against 229E, and this is a coronavirus, so it's comparable to the anti-SARS-CoV-2 titers we having. When they tested the 229E sera against future viruses. So you take a sera of 1980 and try to check on a viral strain of 1990. They found, although the sera could neutralize the contemporary 229E viral strains, they did not detectably neutralize strains isolated 8 to 17 years later. So we can't wait for COVID to, we cannot just sit and think that let's see what happens 8 years later or 17 years later. We have studies that shows that there is a strong antigenic evolution of the viral spike, and what they suggest is that it is advisable to periodically update SARS-CoV-2 vaccines. And how do we do that? So for any war, we need a system of spies. They will tell us and forewarn us that things are going wrong, and this is where you have to attack. For surveillance, we have two very important tools that we can really use. One is genomics, and the other one is proteomics. I will just tell you briefly what happens. You see, in genomics, what we can do, this is a paper again reported in Science, that resurgence of SARS-CoV-2. Hello?
I request everybody else to please mute yourself. Thank you.
Right. How much time do I have left?
Yeah, we still have 10-15 minutes, 10-15 minutes more. Yeah.
Yeah, fine. So what people are doing is, home-based swab collection, and regardless of your symptoms. So even though you have symptoms, you do not have symptoms, you just collect the swab, send it to this national testing agency, who will do COVID first a real-time PCR, and if they find it that they are having you are having COVID, they will sequence the genome. So India also has started. I just came to know yesterday on Twitter that the government of India will start sequencing a few COVID positive patients from every district or every block. So it's a good and welcome step, because this is what these are the weapons of the future that can help us in this fight. So DNA sequencing technologies has really come of age. Ever since the discovery of DNA structure in 1953, uh, now we have brought down the sequencing cost to very low levels. An average cost of sequencing the COVID genome is 8,000 rupees. So it's tracking this. Uh, can everyone mute? I'm just requesting everybody to please
Yeah,
mute
please
themselves.
go ahead. I have muted the person who was
Okay,
speaking.
so sequencing technologies have become more and more high throughput. So on a given day, you can sequence 30 to 40 COVID genomes on a single chip on a next gen sequencer, and it will cost you around 8,000 rupees for one genome of the virus, which is very cheap when you consider the financial and the emotional cost associated with the pandemic. Not only do we have a financial cost, due to closures and lockdowns, we have a huge humanitarian cost that we cannot actually take to pride of. People are dying every day in spite of having the best of technologies available to humankind. So next gen sequencing is a way of understanding how virus is evolving. Much of the data that I showed you today in terms of phylogeny is from NextTrain. NextTrain is enabled by two global initiatives. One is the GIS-Aid. So this GIS-Aid started as a way of tracking influenza mutations. Now they have diversified into SARS-CoV-2. So, and this again, they are part, and then there is this global influenza surveillance and response system that has been working since 1952. And NextTrain, not only do they track the COVID virus, they also track seasonal influenza, West Nile virus, Zika, mumps, dengue, tuberculosis, measles, West African Ebola, avian influenza, all diseases that have a potential to turn into a pandemic. Next Train is tracking. And as you would know by their name, it's called Next Strain. So basically, they started off because they could wanted to predict what will be the next prevalent strain in seasonal influenza. Because in US and many countries, there is there is this system of giving influenza, flu shots, flu vaccines every season. But the influenza virus also changes its strain. So next strain was primarily started off as a group of scientists who would help the government and vaccine manufacturers to predict that which strain of influenza would be prevalent in the next season. Today, they are doing the same thing for COVID. Another part is protein microarray. Now, in protein microarray, what we do is we study the interaction of mm, the antibodies generated in the body against a set of epitopes. Now, this is a very brief figure of how a protein microarray would work in case of COVID. Now, if a person gets infected by SARS-CoV-2, the SARS-CoV-2 will enter into the cell. It will produce, you know, it will, stir the immunogenic response. It will produce antibodies, and the antibodies will be can be IgG, IgM, IgA. In case of COVID, what we have recent research suggests is that there is a very strong high peak of IgA in the initial part of the um, infection, whereas IgG comes later. IgM response is there, but it's not very strong. Now, what we want to do is we want to study the epitope profile. Now, the entire protein is never immunogenic. There is a very specific part of the protein that is immunogenic. Now, if you could collect and study that what are the epitopes that are getting immunogenicity in the current strain. So if we have with for every month, like in the month of June, we collect 500 plasma. In the month of July, we collect 500 plasma serum. Both systems work here for COVID positive patients, specifically for the newer strain, and check if the antigenicity has changed, if the epitope has shifted, then we can certainly predict whether the vaccines that we have already administered will work, or we need to work on a new set of vaccines. Now, these things need to happen at a contemporary pace. It's not that we will study something that happened 6 months ago. We need to hasten the speed at which we are doing our COVID research. It's very fast. COVID is one of the first diseases to get one more than 1 lakh publications for any disease, like if in the publication history of science. So what I'm trying to say is, in case of protein microarrays, we can get strain specific epitopes. So every strain, so in every group one, group two, group three, group four, that are having different strain infection, we can just check if the epitope for each strain is different. There are two things that can be done. If we can get strain specific epitopes, we can design newer vaccines. Also, we can design rapid antibody test. So now we are we're doing RT-PCR. We have to wait for a genomic sequencing to be done. So your RT-PCR costs 500 rupees. Then you take the same sample, spend 8,000 rupees more to get a genomic sequence. That cost could be cut down if we can start working on on protein microarrays and start designing strain specific epitopes and design lateral flow assay systems that can detect those epitopes. Then we can actually identify that, okay, this patient is suffering from UK strain. This patient is actually just suffering from the triple mutant, or pentamutant, whatever mutants are coming in the future. So this is a way where we can use next generation technologies to fight the virus. So let's end this talk with a note. Most of the vaccines that are currently at work prevents the mutants from infecting you. So these are preliminary studies which shows that Covishield, Covaxin again protects against the Indian strain of COVID-19. But are we done with it? No, we need to study more about them, because there's always a warning, because there are recurrent COVID-19 infections. There are multiple studies, there are multiple publications. I could not bring all of them on board here, but there are multiple studies which indicate that COVID reinfection is a reality. Reinfection is a reality even among healthcare workers who had received vaccines and who had always. So, in Brazil, what they found was healthcare workers who had recovered from COVID and got COVID vaccines again got infected with COVID. So, these are warning signs that we need to keep up the fight. We cannot let our guard down, because once we do that, we will end up in a situation uh, where we will have to just wait and watch, just like we are doing today. We are just hoping that the second wave will die down, and it's just hope that is helping us survive today. So with this, I would like to end my talk. specific acknowledgements are due to Professor Sanjeeva. So he actually, instilled in me back in April 2020 that I can work on COVID because I had no history of working on human diseases. I was primarily working on, neuroendocrinology and neuroscience. So I never thought that I will be sucked into this COVID. But he told that he was working with me for another project, and I was there in BHU. He told that you should, repurpose your scientific talent, whatever you have got, and try to help us in this fight. So initially, I was working in. I volunteered to work in the Medical Institute of BHU, IMS. We were doing testing. So we started off with 50 samples a day, and in September, when I resigned from that lab before just coming to IIT Bombay, we were doing like 5,000 samples a day. So I had seen this that lab grow, and obviously Dr. Radhika Vaishnav. There are two reasons why I would like to thank her. First of all, she has been a critique of this slide. So not once but twice. The first time I prepared the slides, I sent it to her. She said it's okay. The second time I updated, again I got her critique. And obviously, thank you all for being a patient listener. I would end with this quote. The scientific literacy is an intellectual vaccine against the claims of charlatans who would exploit ignorance. So the need of the day is scientific literacy, and it's scientific literacy across the board, right from politicians to journalists. Everybody need to be made scientifically literate to understand what's happening. This is not a movie that is going on. This is not a thriller. But once you open the television sets and read newspaper articles, you feel that most of the people have, made this pandemic into a thriller and, settling political scores. This is not the way that we should go ahead. Thank you all. Thank you.
Thank you. Thank you, Dr. Arup. You may please stop sharing the screen so we can have some discussion.
Oh, yeah. Yeah, I'm ready to speak.
A wonderful talk. It's always a pleasure to listen to a researcher. And your last slide was, very. It says the truth that scientific, I think, literacy is the way we can go ahead and fight this pandemic. To start with, I have one or two questions. Uh, your talk about this antigenic drift and shift. Once you said that we are on the brink of a shift, I mean that's little scary. Can you please elaborate on that? Because we are still in a pandemic and we are talking about a shift now. So
No, yeah. So what basically happens is when there are multiple strains of the virus circulating, and when people are okay, I'm getting quick questions. I will take those later. So once we are having multiple strains circulating, and we have this, indiscriminate use of plasma therapy, you are putting a very strong selection pressure. So this virus will try to evade it. So what happens is, just I was telling you while I was giving the talk. Suppose somebody walks into a vaccination center for his first dose, and there are 4 to 5 people. So one is infected with a Brazilian variant, one is infected with a South African variant, one is infected with a UK variant. Now this guy who walks in is not infected. Somehow he's not following the COVID protocols. He's wearing a mask which is hanging below his nose. And many people do that. Like, I mean, 9 out of 10 people I see wear the mask in a wrong way. Even in the, we are when we are in this pandemic. So he gets the infection because most of the vaccination centers are like very close spaces. He gets infected from 3 strains on a given single day. Now, there is a chances of genetic recombination happening inside a cell. So, think of now a cell does not have a single ACE2 receptor. A cell might have 100 ACE2. A single cell is getting infected by maybe 3 variants or 2 variants of the viral genome. Now, when these 2 variants come together in a single cell, there is a high probability that they will recombine. Once they recombine, now you have a variant that has the mutations of the Brazilian strain, the Indian strain, and the UK strain. That might lead to a strong antigenic shift or and what happens is now there is so much of mutation that the existing vaccine won't even work. So, these are, that's why I'm telling that we are the brink. We are the brink of a disaster. We need to stop the spread of the virus. We need to speed up our vaccination. We need to follow COVID SOPs. We cannot just take it too lightly. Now, from India, Indians' perspective, India was fairly successful in controlling the first wave. It was successful because there was a very strong and very stringent lockdown very early into the pandemic. One thing I often say to people is that go by the numbers of infected. What you see, the numbers of infected in India during the first phase of the pandemic, the numbers were not low. We tested very few people. We tested far too less because we didn't have the capability to test. So the numbers of the second wave are higher because we are testing more. If we start testing only the symptomatics, we would again have very less numbers. So if you go to the ICMR protocols, I mean SOPs of getting tested back in May or June 2020, first you had to have all the symptoms to be eligible for getting tested, and you needed a physician's prescription to get tested. Today you can walk into any center and get tested. So now, since we are getting the testing done more at a much rapid pace, we are finding more infections, so we have higher numbers. So but even then the first wave was less because of the stringent lockdown. People were kind of afraid. Now people are superheroes, so they go around without mask. In the first part of the pandemic, people were actually afraid. Then people thought कि नहीं, कोविड से तो लोग उतने मरते नहीं हैं, कहाँ लोग मर रहे हैं, things are normal. By September we had hit the peak, and then by Diwali everything was gone. People were like, there were farmer protests, there were political rallies, and everywhere people thought कहीं पे तो COVID हो नहीं रहा. And we were caught napping. Second wave, we were caught napping. The general public, the politicians, the bureaucrats, and I won't blame any single group of people. This our entire society was caught napping, and there is no point blaming a specific government here and there. We were caught napping, and that's where we are here. So anyway, maybe I've digressed
Uh,
from the topic.
Dr.
So
Arup,
antigenic shifts and antigenic drifts are happening because more and more people are getting infected. More and more variants are into, in
right.
circulation already. So
A quick take on plasma therapy. You
right,
said plasma therapy is to an extent responsible for this, uh, shift and drift. So,
right.
what percentage of population is required to take plasma therapy to really make an impact on these, drifts? Because I don't think so many people
Uh,
are getting plasma therapy as to impact the whole scenario.
when you talk of percentage of population, let me tell you, this pandemic perhaps started with a single person back in 2019, and in 2 years we have billions of people who are getting infected.
Right.
So what happens in plasma therapy is, suppose I get infected with a variant, specific variant. Let's call it variant A. Somebody is infected with variant A and B. Now, one more thing, we often talk of variants in terms of like we are getting only one infection. Sometimes we get multiple infections. So the viruses we breathe in the air is not just one variant. Now, during plasma therapy, just like antibiotic resistance, you are applying a selection pressure. You are allowing few spike mutations to die down, whereas few spike mutations are not dying. So I have antibodies against the spike protein. So those spike protein antibodies will neutralize in the patient to whom I'm donating plasma, will definitely neutralize those spike protein mutations which I already had. But it cannot neutralize a section or a subsection of those viruses whose whose spike has already mutated.
Right.
Now, once the old variety of the virus has died off, now you have a select bunch of viruses which are now resistant to my plasma. Now they will rapidly, multiply, If it is an immunocompromised person, somebody who had undergone a chemotherapy, somebody who had a transplant, and these people should never be given plasma therapy. People who have immunocompromised status. I don't know what is the status in India, but I see quite rampant use. And I don't even blame the doctors, because somehow two fake stories have been planted in the person's mind. First, remdesivir saves your life. It does not save your life,
True.
but people believe remdesivir saves your life. The second fake story is plasma therapy saves your life. That's why I was talking of scientific literacy in my last slide.
Right.
People need to be, scientifically relevant, and journalists and people who are spreading this entire mass media thing, they need to talk to actual scientists. People are talking with Twitter influencers on a public forum and discussing medical things that only doctors and researchers can deal with. So that is a problem. And you have also researchers who will, I mean, the same group of people will discuss on budget. The same group will discuss on, elections. The same group will also discuss on vaccination scheduling. The same group will discuss on plasma therapy. Now, this is ludicrous. At least have specific people for specific tasks. But in India, you have, once you get a blue tick on Twitter, you are an certified expert of everything. So that becomes your
Coming
job.
coming back to genomics,
Right.
quick few quick answers because I want some other people also to interact. Uh, my question is regarding genomics. How
Right.
little we are doing as in India, no, as a country. How far? I mean, we are lagging behind in terms of doing genomics.
In India, we are pretty far behind. We were actually very pretty far behind, because India did not have this mandate to do the sequencing. But after the advent of UK strains, and when people started talking of the advent of, I think UK strain was the first big knock on our heads that this virus is mutating in very weird ways. So after these things started happening, India has started sequencing in India has gathered pace. And I was just saying, like, the other night, like, just yesterday night, I don't exactly remember the specificity, but it so happened that I just studied that Indian government has now talked with IGIB and CCMB to sequence from every district, and that is the way to go ahead. You have to sequence periodically, every 15 days. You have to sequence from each cluster, and we have to look out. We have to talk with epidemiologists and public health expert and look out for breakouts.
Right.
This second wave kind of started in Bidar, Maharashtra. Uh, and again, uh, people have to talk with scientists. I mean, you have to talk to the right person for the right advice. It's not that the same person can give you all the advice.
So, will these
But
genomic studies help in designing new vaccines? I mean, are our
yeah,
vaccines
surely,
amenable
surely.
to redesigning, like say Covishield or Sputnik? Are they amenable to redesigning, or it's only
Yes,
mRNA?
that's no, no. Even Covishield and Sputnik are amenable, because you see,
Okay.
they have not relied on proteins. They
Okay.
have not relied on proteins. They have relied on the genomic information. That's why they are pretty new generation vaccines. We did ordinary vaccines that we take, that we have been taking. Those are, we have never taken an mRNA vaccine. This is
Right.
first time an mRNA vaccine has been used, and the adenoviral vector vaccines also are very new. So these are obviously amenable to change.
Right.
They are pretty fluid and plastic. That's the reason in the first case they were approved. So what Moderna has to do today to get a new variant, to get a new version of the vaccine, is just change the genetic code. You don't have to change the entire delivery system. So nothing needs
And
to
of
change.
course, they will not have to go through all the phases of routine vaccine development. I mean, they can
Uh,
be
um,
translated into use within few months' time.
yeah, sure. Because you see, the delivery system is different. The genome sequence is different. So it's just changing the information in the genetic code. So that's why mRNA and adenoviral vaccines um, can be updated, in a jiffy. You don't have to wait for 2 to 3 years.
Right.
But as of now, I would say, the given the pandemic and given the slow pace of vaccination, a vaccine such as Covaxin is much more practical. First of all, it kind of exposes our immune system to all variety of viral antigens. So although your humoral immune response against the vaccine, that is the neutralizing response would be less, your T cell response will be pretty good, especially in the younger generation or the younger people, 18 to 45, 50. That generation of people would really give a good T cell response as well. Even the older people can, and that is what we are seeing. Many of cases, people are checking, people are getting their first dose and getting infected with the virus, maybe after 1 month, after 20 days, 15 days. And what I was going through a paper, I had so many papers to share, but time is a limit. So I was going through a paper where they show people who are getting their first dose are also showing a strong T cell response that is enabling them to clear the virus at a faster pace than compared to people who had never been exposed to the virus.
Right.
So this is there. So we need to believe in our immune system, both the B cell neutralizing antibodies and as well as the T cells.
Thank you. Thank you, Dr. Arup. Now, I invite participants to interact. I see Dr. Srinivas Kasi, who is a very eminent vaccinologist from Bangalore. Dr. Kasi, if you are hearing, can you put up some comments? Dr. Kasi, are you hearing?
He has actually left a question.
Okay. Okay. So you
No,
may
no, no.
address this
Yeah,
question. Yeah.
I'm here.
Yeah. Dr. Kasi, any comments on what Dr. Arup has said? Anything to add or your query on that?
Well, one query is that whether the changes that have been detected so far, is it time to go for a new vaccine right away, or do we still have some time with the older vaccines? And the second is, this talk has been an eye-opener. We have never heard anything like this in the forum of pediatricians. And I would like to information.
Yeah. So the question you're asking is a very pertinent question. Do we need to update our vaccines? Perhaps in the next 5 6 months we really need to. Now the problem with COVID, it has not been a typical disease. I think all of us would agree on this. It has become a socio-political, a geopolitical, you know multiple big farmers have invested a lot of their money in developing vaccines, and now you see there's vaccine politics going on. Other Punawala had to get Y category security. I mean, what kind of country are we living in? The guy who is supplying us vaccines needs his security to be upgraded. So the question whether we can upgrade the vaccine, yes, ideally, in a very ideal case scenario, we should update our vaccines. Uh, I would love to see AstraZeneca, and what we can do actually is make a cocktail rather than just giving a single spike. We can make a cocktail of existing mutations and make different spike protein variants and give a cocktail of vaccines. But I think, given the limitations of the pandemic, once you have a newer version of a vaccine, the will the older version be acceptable to the general public? I don't think many people will will go into another round of vaccine hesitancy. As of now, AstraZeneca is showing a success rate of around 70% plus. Uh, which is kind of good. People are getting mild infections, but maybe in the next 1 year we need to update. That will become a necessity. Now there are lots of it's a not a question of scientifically. Yes, I would say yes. You should update. You should update it every 1 month. But will it be feasible? It's not even a technological non-feasibility. Technologically, it's feasible to update it actually every 3 to 4 days. But when it comes to mass scale manufacturing and build this obsolescence, now if suppose people get to know that now AstraZeneca has Covishield version 2, what happens to the billions of doses of Covishield vaccine version 1 being manufactured by Serum Institute? Nobody will buy it. People will nahi nahi mujhe naya version chahiye. Just like we run after the new smartphone, just because the new smartphone has 88 megapixel and mine has 82 megapixel. So we would try to get that 6 megapixel advantage, and we run behind the new model of the mobile phone. So these are commercial questions. I don't know when companies will open up to these questions. As of now, Covishield is working. It is exposing you to a wide gamut. You see, it's around 3 kb of viral genome. So although I showed in this figure, I would just like to show you this. I don't know. Can I start screen sharing again?
Uh,
Yeah.
no, but we will be running short of time now. So
Just I just wanted to show one figure. Yeah. So is this visible?
yes,
So if you see, the spike protein has mutated pretty high. What you see amongst all the genomic regions, it is the spike protein that has the highest level of entropy. So I guess spike protein based vaccines need to update itself. Maybe in the coming 6 to 7 months, maybe they would do that. But as of now, we have to stick with the vaccine that we have got, because vaccines take some time to develop. These are the fastest range of vaccines that have been developed. I think we can stick to it. The something is better than nothing. That's the tragedy of this pandemic.
right. So there are some questions. They are very routine questions that we hear in all the forums. Would you like to answer those quickly?
Yeah, maybe I
Quick
can give.
fire. Yeah.
Yeah,
How much
quick.
probable is doctor who are vaccinated in first phase will get infection now?
Depends on the strain you are exposed to. If
Right.
you're exposed to older strain, yes, no, you won't get infected. And but even if you get infected, there is a fair bit of chance that you never would have the severe form of the disease. You will have a mild form of the disease, and in a good way, your immune system will get a booster dose, a triple
Right.
booster kind of a thing.
Then Covishield is available to most of us as of now. What are the chances of protection with present Covishield vaccine with the kind of mutation that you are described? I think that's the same question. Would you
Yeah,
like to
I
comment?
think we dealt with this.
Can you share something about triple mutant? You already shared it, I think, during the talk. You talked about this triple mutant.
Yeah,
And then what about booster dose
with
with either
Covaxin.
Covaxin? Yeah, with Covaxin, that is what.
No, I don't think we should go for a cocktail party. If you are getting an AstraZeneca vaccine, get the second dose from an AstraZeneca vaccine, because that is how vaccines work. Your immune system will be kind of perplexed that पहले कौन था दूसरी बार कौन आया, so it would end up nowhere. So if you got a Covishield, stick to Covishield. If you get to choose between Covishield and Covaxin, I would ask you, with my knowledge of science, that you can stick. If you can go for Covaxin.
Right. So, what about future prospects of having a booster dose of the same vaccine? Say because the immunity will wane. They say about 9 months, 10 months. Then
Yeah, I think I don't know if the government will allow you to get a booster dose, given
hmm,
we have around 140 crore people to get immunized.
hmm,
But yes, if you can, even after 5 6 months, the markets open up and then the vaccine availability goes up. I would suggest that every 6 to 7 months, I think even I don't know if doctors and people who are actually in charge of these things understand human body much better than us. Like, we are scientists, we get only the science of it. We don't get the clinical aspect of it.
true.
But booster dose, I think, won't be a bad thing, because just like influenza, and I just showed you that every 2 to 3 years we tend to get infected by the same coronaviruses. Uh, so our immunity wanes. But new studies are showing that, since we didn't have much time, new studies are showing that even after 9 months, there is considerable viral titer. But again, there are studies that shows reinfection. So there are all kinds of
Right.
studies across the board.
Right. So,
We
any more
have to,
questions from any
use,
comments from other people? I mean, if somebody wants to ask question, we will spend about 3 4 minutes more now before we end the meeting. So, any more questions or comments? Yeah, it seems there are no more questions. So, you were saying something and I interrupted. Sorry. So you can restart, Dr. Arup.
No, I think what I was telling is the best way of vaccinating ourselves is using the mask properly, using the COVID, following the COVID SOPs. I know it's very tired. We kind of, pandemic fatigue has set in. Most people are like tired, like और कितने दिन घर में रहेंगे? How, like, we want to party, we want to do this, but you see, it's a long drawn battle. Even if you feel sad, think of those people in Europe who had to endure World War II for almost 5 years. Think of the people, like every day they had to rush to bunkers. They didn't have any access to water. They didn't have proper access to food. They didn't have proper access to like anything, and they were bombed. Ultimately, after like think of yourself in a war zone. What would you do? Save your life. And this will be a protracted war. This will keep on going. Uh, we have to keep faith in our, keep hoping that things will go for the better. The the first chance you get to get vaccinated, do get vaccinated. Don't as scientists and doctors, we should dispel the fears. If you see, I was just going through old news reports. There was so much of vaccine hesitancy in January, and people were trying to make it a political slugfest. So I think people across the board should avoid these things. We are dealing with a pandemic. This is a public health disorder. Let us not make it a political slug fight, because we need to support all our local governments. Whatever government is serving you, support them. Try to. You can complain, but overall, there is no fight, no advantage if we start bickering with each other. And that would be my honest suggestion. And obviously, I don't know when journalists will learn, but most of the reporting on COVID has been pathetic, to say the least. This is not a T20 match that you need to sensationalize everything.
Right. We had, in
So,
fact, Dr. Naveen Thakkar about a couple of weeks back, who talked very nicely about how to fight vaccine hesitancy. So
yeah,
once again, thank you, Dr. Arup, for enhancing our scientific literacy. That is what the Saturday Club of Baroda is meant for.
I'm
And thank you
saying,
so much for your time. And we'll definitely have you again sometime later with some more advances in the field of COVID vaccines. So thank you so much
thank you
for
for
joining us.
hosting me, because this is a very old club. Dr. Radhika told me that this
Yes.
has been going on for the past 50 years, and I was
Yes.
really blown away that how could something
Thanks
run for
to
so
thanks
long.
to one Dr. Satish Shah, who is urologist, Dr. Dakshaben Shah, pediatrician, Dr. Amin, who is a pathologist, and Dr. G.N. Vaishnav, who is an internist. They are all into their 70s or 80s, and still you can see them really on board just now also. So they
Yeah,
are the people who have been carrying on with this activity. I have taken a baton to carry it on online.
yeah. So thanks to all of them, and thanks for this. People should keep on learning, and knowledge keeps on expanding. Obviously, I would like to join you guys again, and hopefully, maybe give you some new news on
Thank
how
you.
we are going on with this pandemic. Stay safe, everyone.
Thank you so much. And so
Thank
I
you.
thank everybody for joining, and we end the session here. Thank you so much.
Thank you, sir.